Hello, my name is Graeme McDowell, and I'm with the National Research Council of Canada, here today to talk to you about my poster titled Computational Membrane Lipid Structure Inspector, or CoMLiSI, a tool to verify lipid structural elements in PDB files. First, I would just like to say, because I don't have much time, I will be going through this rather quickly. If you wish, please pause the video so you can read any of the text that I don't get to. So first things first, the computational modeling of biological membranes is a field, a rapidly expanding field that allows for the study of molecular and cellular processes, getting new insights into disease states, as an example. But accurate models of the components of these in silico experiments are crucial to maintain accuracy in the simulation results. But manual curation of these structures is extremely taxing and error prone. So while the structure files, either PDB or GRO files, contain atomic information, including the atomic coordinates, they do not always include bonding information, and so errors can occur during these simulation runs. So we developed CoMLiSI to assist researchers by verifying structural features of lipid molecules within the membrane structure files. So here in figure 1 is just an overall flowchart of how the program works. So first, the user uploads a file, clicks run. The file will then read and parse through, just retaining the lipid molecules. It'll calculate the lipid features, check for errors, and then repeat that until all the lipid structures are finished, at which point it will then collate the membrane features, the membrane in this case being the file that is uploaded. And it will populate an R-Shiny UI showing in the membrane tab visualization of the membrane lipids, a scatter plot showing where all the lipids are in 2D space, and summaries of the lipid membranes, so including which ones have errors, which ones have cis bonds, trans bonds, etc. And in the lipid tab, we have the visualization of individual lipids and a summary of these lipids. Lipid bond histograms, as we can see down here, and I'll talk about that in a moment, and then various adjacency matrices. So during the process of running this program, first it parses the lipid structures from the structure file. So if you have a file containing hydration, ions, protein, anything like that, it will remove those and ignore them in the processing step while uploading the file, and will then look at each lipid structure individually. CoMLiSI will then calculate the lipid features. So this includes bonding. So for example, it'll calculate the distance between these two carbons here and determine that's a single bond. So it will draw it as a single bond. In these two carbons here in purple, it calculates that those two are within the distance for a double bond. So it saves that as a double bond in memory. And it will do that for each atom and connections between each atom in each lipid. Once it has the bonding pattern, it will then look at other features. So for example, it'll calculate the cis or trans isomers or configurations rather for lipids. So in this case, it will look at the distance between the hydrogens on the two double bonded carbons, as well as looking at the dihedral or torsion angles between them. So in this case, the hydrogen, carbon, carbon, hydrogen, or this carbon, 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 carbon chain. If the dihedral angles and the distances all match up, it is confirmed to be a trans bond or a cis bond, depending on the values reached. During the error check, the program looks for various discrepancies in the structure. So for example, here we can see there's a carbon that has 3 hydrogens attached to it, as well as 2 other carbons, which is too many bonds for most carbons that don't have a charge to them. So in that case, the program will flag this as an error and show and display that to the user. In this case here, we see there are 2 carbon-carbon bonds, 2 double bonds. One of them is a cis bond, and the other one comes out as an error because the distance between these two happens to be within the range of a single bond, not a double bond. So this comes out as an error. The user can then do with that as it will. In the UI population, the membrane tab, for example, here we can see this is a representation drawn in NGL Viewer on all of the lipids within the file, and you can highlight certain aspects, so the lipids containing cis bonds or trans bonds, etc. And the bond length histograms we can see here, as I mentioned earlier, these are histograms showing the collection of all of the various bonds within the lipid. So at the bottom here we have the individual lipid that's selected, in this case PIPI 194, and you can see all the cis bond, all of the carbon bonds, and then the carbon-oxygen bonds here. You then will show all of the membrane bonds themselves, and then a reference membrane that we generated. So using this program, we wanted to see how it functions in real-world databases. So here we took 16 membranes from the S-Lipids database and 6 from the Limonada database. Now, for each one of these, we know what lipids are in each one of the membranes, and these lipids have defined structure. So we marked if there were any lipids within those membranes. So for example, in one membrane in S-lipids, we found that there is a discrepancy between the cis and trans configuration. So for example, we know that there's supposed to be a cis bond, but it ended up either being a trans bond or not there at all. So in that case, we have one membrane that had at least one or more lipids with this error in it. So as you can see, even in these publicly available databases, there are still lipids within their membranes that have some of these errors that crop up. So while the number itself may be small, it is throughout, which implies that caution is required in order while using these databases or generating those of your own, in order to see, are these structures accurate? And so just to conclude, we presented new software here that's able to parse out the lipid structures, calculates features of them, and returns information of those to the user. And then using this software, we showed in publicly available databases that errors and discrepancies can occur. And so caution must be used while generating lipid membranes for simulations to ensure that the structures are all accurate. And we believe that CoMLiSI can be an integral part of structural curation for these membranes. Thank you very much, and I'd love to answer any questions if you have them.